Biotechnology can be defined as the use of microorganisms, plants, and animal cells for commercial scale production of agriculture, biopharmaceuticals, and anything which can be helpful for the human welfare. Application of biotechnology in the field of agriculture is very evident in and is responsible for most of the changes which took place after Green Revolution. The three options for increased food production can be agrochemical based agriculture, organic agriculture, and genetically engineered crop based agriculture. The Green Revolution actually succeeded in increasing the yield of the crops, mainly due to the improved varieties of crops and the use of agrochemicals, that is, the fertilizers and pesticides. Now, with the use of more agrochemicals like these fertilizers and pesticides, the pollution rate in case of soil, water tend to increase, and because of that, the overall environmental hazardous situations kept on increasing. So, the biological agents can be used for sustainable development in the field of agriculture and giving very good yield. So, keeping all this in mind, we have to discuss some of the points or some of the advances which were which are taking place or which have already taken place in case of agricultural field. We will be discussing about some of the GM plants, that is transgenic or genetically modified plants, and how are they helpful for the farmers. Now, first of all, we should understand that what is the meaning of stress. Plant stress is a state where the plant is growing in non-ideal growth conditions that increases the demand made upon it. The effect of stress can lead to deficiencies in growth, crop yield, permanent damage, or death if this stress exceeds the plant tolerance limit. Anytime when a plant is kept into a condition which is not normal or natural to it, there is a chance that the plant may start releasing certain hormones. And because of the release of these hormones, the growth of the plant will degrade and ultimately stop, and it will lead to the death of the plant. This can happen if there is climatic change which is occurring very fast, and other than that it can be use of these fertilizers and pesticides as well to the soil, which can completely disrupt the normal functioning of the or normal growth of the plants. So for the basic idea, when the crops they were thought of to become, or the scientists were thinking of making genetically modified crops, they always thought of various conditions. Like some of the plants they only grow in colder regions, some other they grow in only temperate climatic conditions. So there is a gap for the production of these plants. The plants which grow in temperate conditions, they cannot grow in the colder conditions. So there must be something within the plants which grow in temperate conditions or in cold conditions, and that is lacking when the situation or the environmental condition is changed, and that's why the crop is not growing properly. In case of oranges, for example, you can very well understand when you go to Kalimpong side, you can very well understand that the plants they are having or very sweet tasting oranges, whereas when we come to the plain side, even in the doors we can see the oranges growing, but those oranges they are sour. The reason for this is the environmental condition in which the plant is growing. So the scientists they kept into mind these all climatic conditions, and these all are stresses. Stress means any condition which is not natural, like cold, drought. They are natural, but they do not they hamper the growth of the plant. That's why we call it as abiotic stresses. So abiotic stress such as cold, drought, and salt, heavy metals largely influence the plant development and crop productivity. To cope up with these abiotic stresses, plants can initiate a number of molecular, cellular, and physiological changes to respond and adapt to such stresses. Sometimes these stresses can be combated with the help of various strategies. You may have seen that in the roads, when the roads are completely filled with snow, the like people, they sprinkle salt over it so that it melts. Similarly, the plants also have certain metabolic activities going on in the body, so that accumulation of salt or glucose or sugary substances take place in the body, and this will help in fighting against cold. So, in temperate latitudes, the term most often describes resistance to cold or cold hardness and is generally measured by the lowest temperature a plant can withstand. Hardiness of a plant is usually divided into two categories: tender and hardy. Transgenic rice plants expressing CSPA and B manifest improved stress tolerance for a number of abiotic stresses like cold, heat, water deficit. It has long been established that changes in gene expression occurs upon exposure to cold acclimatation. It means the plant is getting tolerant over cold climatic condition. These transgenic plants, they are modified by changing a certain gene within them by introduction of with the help of agrobacterium tumefaciens or some other method. Now, nutritional enhancement in crops, it targets manipulation of levels of proteins, amino acids, fats and oils, vitamins and minerals as well. Even sometimes carbohydrate and fiber content is tried to be increased. Sometimes, like presently, there are various varieties of rice which are formed, and they have very high, like golden rice is one of it, and it has very high content of vitamin A and beta carotene. Similarly, banana varieties are formed which have very high level of minerals and vitamin within it. We can modify any of the plant or fruit by introducing certain genes of particular uh, desired variety and manipulate the growth of the plant, and just by consuming that fruit you can you will get the same value what a tablet will give you. So this can be thought of in the near future. Pest resistant crops are also there. Till now, we have discussed only the varieties which were giving good yield or high nutritional value. Various other plants are also there. I just mentioned about golden rice. The pest resistant crops, how are they formed? Again, pest is, we have to understand the physiology of a pest. How does it attack the plant? And what can be done in order to not allow the plant to be infected by the same pest? So, for understanding this, we need to understand the life cycle of the plant as well as the life cycle of the pest, even the feeding habit of the pest. If we can understand this, we can definitely manipulate the plant in such a way so that we can get our desired pest resistant. BT cotton is one of the genetically modified crops, and Bacillus thuringiensis is a bacterium that produces an endotoxin. This endotoxin or like the soil bacterium Bacillus thuringiensis, when it produces these cry proteins which are endotoxins, they are toxic to the larva insect of tobacco budworm or army worm or beetles, some mosquitoes as well. The cry proteins, they exist normally in an inactive protoxin form and get converted into active toxin when they are ingested by the insect. As the alkaline pH of the gut, it solubilizes the crystals. When the activated toxin, it binds to the surface of the epithelial cells of the midgut of insect, it creates pores or holes in the gut of the epithelial cells of midgut, and because of that swelling develops. And as the swelling develops, there is lysis of cells, epithelial cells, and this leads towards the death of the larvae. Now, the genes which encode these proteins can be isolated from the bacterium, that's like Bacillus thuringiensis, and it can be incorporated into the plants like cotton, tomato, corn, rice, soybean, and various other plants. And with just the manipulation of this cry protein, in order to form the transgenic plant, we just need this cry protein to be formed. And cry gene we have already identified. Our intention is to incorporate this cry gene into the plant, bt cotton plant, and after the plant develops, we find that every part, even the leaves of the plant, has the presence of this pro- protein. So cry will be there, cry will produce the cry protein, and as such, the entire leaves of the plant has the presence of cry protein. Now, whenever the bollworm larvae is feeding over the plant or bt cotton, the larvae will have pores in the gut, midgut, and they will die. And eventually, the plant will be saved from the pest attack. So, this was the idea. Now, there are various types of cry proteins which are introduced into the plants in order to fight against different kinds of pests. For example, cry 1AC and cry 2AB control cotton bollworms. Cry 1AB corn borers. Cry 3AB controls Colorado pests. Now, this Colorado potato beetles, they are controlled by Cry 3AB gene, and Cry 3BB corn rootworm controls the corn rootworm. I'm sorry, Cry 3 capital B small b gene, it controls the corn rootworm. So these four you have to learn, and it is very important. It is asked many times in the examination. This was about the pest control. Now, next is about the RNA interference. What is the meaning of interference? Interference means hindrance, right? So the idea is the same. Now, RNA interference technology is used to block the expression of certain genes and it also allows gene silencing. Gene silencing means suppression of a particular gene. During this process, a complementary RNA and to the mRNA being produced by the gene is introduced into the cell. For example, you all have an idea that when transcription is taking place, we expect that only one of the suppose this area will only be capable of forming an mRNA, but the below area, this area doesn't form the mRNA. What if this area forms the mRNA? If this area will also form the mRNA, then we will get something like this, and there will be production of a double stranded structure of mRNA. And when a double stranded structure of mRNA will be formed, they will join together and they will not be functional anymore. And because of this, there will be no activity